residue in plant cells. So using plant cells as a source for proteins. The advantages of this technology are many different ends, and just a snapshot where we are today. We have our first late-stage project for development for an enzyme for treating Gaucher's disease. This protein is under FDA review. We'll of course say a bit more about where we are with this lead protein in a moment. We have a number of additional proteins in different stages of development, and perhaps I'll be able to say a bit more about these today. We have a partner for our first product, which is Pfizer, who has the worldwide rights for marketing our Gaucher's disease product, and we are using our platform for production and development of many other different therapeutic proteins I will run through quickly today. So first, just to give you a real-life picture of how it actually looks, these are the bioreactors, the polyethylene base, where the plant cells, the parent cells are grown. All this is done within a clean room environment, so all the regulatory expectations from the different authorities are implemented in the production process, and indeed actually our site has been approved by FDA recently. On the other hand, we are using a novel type of cell. So these are plant cells, not mammalian cells, not bacterial cells, but plant cells who are producing the human therapeutic proteins. Our most advanced protein is plx-israel alpha, an enzyme we are developing for treating Gaucher's disease. This is a genetic disorder which is actually more highly prevalent among the Jewish Ashkenazi populations. So actually even Israel is a relatively significant market. The lack of the enzyme causes accumulation of a substrate that is accumulated in the spleen, the liver, the bone, and the lung. And in severe cases, these patients, if they're not treated, unfortunately ultimately die. This is a unique market. Only around half the patients are actually treated due to primarily diagnostic and financial constraints. This is an orphan drug on one hand. On the other hand, the average treatment per patient is about $250,000 a year, which brought in the peak of sales for over $1.25 billion markets in sale. Currently, there are two drugs in the market, Cerazine by Genzyme and Aviezer by Shahar, both made in the mammalian system. So, with this enzyme made in plant cells, we went down to a development program. This is not a medical meeting, so I won't go too deep into the medical details. Basically, the endpoints we're going after are the classical endpoints developed previously and accepted in the medical community for treating Gaucher's disease. We're talking about, again, decreasing in spleen size, decreasing liver and elevation in hemoglobin and platelet count. This was agreed upon with the FDA under an SPA agreement. Again, the classical endpoints. And as you can see, for the primary endpoint, we used two different doses. We hit the endpoint already after 6 months, demonstrating the efficacy of our protein made in the plant cells, and going on to the additional endpoints such as elevation in hemoglobin. And we can see in those anemic patients, the elevation is dramatic. In the two doses, we can see a very clear increase in hemoglobin count, a decrease in the liver size, and again, those with hepatomegaly, the decrease is even greater, again demonstrating the efficacy of the drug. Same here for the platelet count, again, a clear efficacy with the protein. As for safety, here too, the pivotal trial had no serious adverse events. We had no neutralizing antibody formation. We had 6% had developed antibodies to the protein that were not neutralizing. This is even lower than the rate expected in the literature, and around 6% with hypersensitivity, which is in the same range of the mammalian expressed protein reported. So overall, the safety data here too for the pivotal study, together with the efficacy, is very compelling. Nevertheless, we thought it was important to demonstrate that not only can we treat naive patients very successfully, and we already have naive patients on treatment for already 3 years, but also to show that we can take the patients who are stable on the mammalian cell expressed protein, Cerazine, and transfer them to be treated on our drug successfully. And this switchover study was a a relatively newer study that we have initiated. It was divided into two cohorts. This is starting out with 9 patients, with 25 adults being treated under this protocol. What we can see, and again very briefly, is a very clear picture of stability. Again, these patients were first demonstrated that they were stable on Cerazon, and only then have been switched onto our drug. And you can see for platelet count, a very clear and stable picture. Same goes for hemoglobin count. You can see that the patients maintain the same level of stability when transferred from a mammalian cell protein to a plant cell protein. The spleen volume as well, we can see the level of the spleen remains the same. You can see a very slight decrease, which I don't think is very significant. But we can see a very clear picture here as well. So also on the organ level, we continue to see, this is true for spleen and liver as well, we can see the continuous stability of patients treated for at least 2 years, and some have been even much longer, on the mammalian cell expressed protein and when they are transferred to the plant cell proteins. This is a very important data, because actually, when we look at the real world of where we are aiming for treatment, the first majority of patients will not only be the new naive identified patients, but the thousands of patients who have been treated before are definitely a group of patients we would like to be able to treat as well, switching them to our drug. So this new important data is very important as well. Furthermore, we followed a very important parameter for the Gaucher patients, which is bone. This is an MRI analysis of the quantitative chemical shift imaging that measures the fat fracture of the bone marrow. And you can see green is good in this case, no green is bad. And you can see that these patients, you can see their score going up dramatically from what is demonstrated as bone at risk all the way to healthy bone within 24 months. You can see this is a very clear picture. And this definitely seems like something that is perhaps a unique property for our enzyme, demonstrating very good efficacy on this important endpoint, which is bone treatment for these patients. So, all in all, this data has, of course, working closely with the regulatory agencies. We have submitted our NDA filing some time ago. In February of this year, FDA asked us to gave us a complete response letter asking for additional data, primarily around the switch data. They did not have the switch data in the NDA file. They have understood that indeed this data is important data they would like to see. Therefore, they asked for this additional data. We have met recently with FDA, and we've fully clarified with them that the data that they want is data that we have. So we are now, there's no need for any additional clinical trials, any additional follow-up. What we are doing now is compiling this clinical data for FDA in order for FDA to be able to have, first of all the switch data that they did not see before, and furthermore updated efficacy data from the naive patients as well. As I said, we have patients going all the way to 3 years of treatment. All this data, together with some additional CMC points that FDA asked us to clarify, again, data that is in hand. So we expect to be able to submit the additional data FDA requested in the next few months in order to enable a rapid evaluation of this data by FDA and looking at approval hopefully still this year. So this is where we are with FDA. Rest of the world, we have submissions going on in addition to several countries. In Europe, where we just received the Day 120 questions. We have an application in Brazil with Aviezer, the Brazilian regulatory authority, that is expected to come very soon to audit our facility. So we have 2 authorities who already have performed their audit, which are the FDA and the Israeli Ministry of Health, and the additional authorities as part of the review process will be coming in to perform this